So for the remaining of the day, we are going to go into macromolecular docking. You've been hearing this morning about small molecule screening and docking using the HADDOCK tool developed here at academia. And I'm going to tell you about information-driven modeling of biomolecular complexes. So we'll have a lecture this morning until about 12:30. And this afternoon, you're going to do some work, actually, using the virtual machines. You have hands out of the tutorial where there are quite detailed instructions to run things. It's only a fraction of the tutorial that we are offering, so there are many more tutorials there. Well, you can see probably from the content. So what I'm going to tell you today, I will give you first a short introduction to HADDOCK. Well, maybe not so short, but some introduction to HADDOCK. I will not speak in general terms about docking. I will assume that you know enough of that. Then I'm going to describe the modeling of larger assemblies, meaning by that systems that are built of more than 2 molecules, and also how to deal with large conformational changes. There are a number of challenges when it comes to modeling complexes, and those challenges are the energetics, so the scoring of solutions, how do we know if a solution is good or not? And the other main challenges in this field is if the structure of molecule changes a lot when it's binding to its partner, it's very difficult to model those. So we are very limited in what we can do. So I'm going to speak about large conformational changes, mainly domain motions. I will show you a little bit of example on incorporating biophysical data. Actually, not so much. For those of you who are from Academia Sinica and not following the ISGC conference, I'm going to speak tomorrow in the biological chemistry department, where I'm going to tell a little bit more here about.
It's 11 o'clock on the first floor of IBC, Institute of Biological Chemistry. Alexandre will talk about integrative modeling.
So I'm going to speak there about things that I'm not speaking here today about. So it's covering. Although in the slides that you have in the handout, there are many more slides that I'm going to speak about. So I think if I was to handle all the slides that you got there, I would be speaking for 3 or 4 hours. So we're not going to do that. Then you will have to make a choice what you want to hear, because time is limited. So I have different topics. So we'll make a vote, and then we'll see what you want to hear. And I'm ending up with challenges. So as you're all very well aware, we have a huge amount of genomic data that are being generated and that are generated. We have hundreds of genomes that have been determined. And going from a genome, you go to the proteomes that are the express protein from a genome. And then if you go one step further, you get into the interactome, which is the network of interactions in a cell, basically. And that's a highly dynamic system. You see those maps. These are typical 2-dimensional maps with a lot of dots and a lot of connections. Many more connections than dots. On average, from estimates of different interactomes that have been mapped, each protein in those systems interact with 9 to 10 other molecules. So lots of interactions. Those maps are also dynamical. It means they might change as a function of time or as a function of where you are in a cell cycle. They will also change as a function of post-translational modification, so there's a lot of regulation. So it's a very complex system. So it's by far not 2-dimensional, but you have to think of it as a 3 or 4-dimensional system, where time is one dimension, location in the cell is another dimension that defines the fate of interactions. And if you want to add the structural dimension to those networks and try to understand what is going on in those networks, you need to model at some point those complexes or solve them using structural method like X-ray crystallography and NMR. But because there is a huge amount of them, and some of them are very weak and transient, they are not simple to study by NMR and X-ray. And you also have a lot of those interactions will be involved in membrane and membrane-associated proteins. And again, these are challenging to study by NMR and X-ray. So the methods there to complement the experimental structural method will be docking, which is the prediction of the structure of the complex based on the structure of the constituent. And if you have that, if you have structure of complexes, then you can think of drug design or screening maybe for small molecules. There's a lot of research these days also trying to find inhibitors of protein-protein interactions. But this molecule will have typically, or most likely, different properties than the small ligands that are used now as drugs. Because those interfaces are they don't have well-defined binding pockets. So the properties of the molecule will be different. Most likely they will be slightly larger. So in the previous sessions, there was this rule of 5 that was mentioned, the Lipinski rule. If you look into protein-protein interaction inhibitors, you will have to relax those rules. Otherwise, you will probably not find molecules that are able to inhibit those interactions. They will be slightly large. And given the structure, then you can also think of going toward affinity and predicting affinity from structure, but this is still an open challenge, and I will end up with that part in my lecture. So I basically mentioned all of that. So docking is really the prediction of the structure of a complex based on the structure of this constituent. And actually, if you are interested also, there is an experiment, a worldwide experiment, where we are measuring the state of the art in docking. So where are we standing? And groups worldwide are participating to this experiment and trying to predict the structure of complexes. There's also a lot of work in bioinformatic predictions of interactions, and there are groups here at Academia Sinica who are also very active in this field. So what you are going to get out of this kind of modeling are not hardcore 3D structures, but models. And there is no clear distinction where the structure starts and when the model ends. There is no clear definitions of how much experimental data do you need to get to define something as being a structure. But those models can provide insight into function and mechanism, of course, of action. Very importantly, I think the model should not be the end of the research line, but the model is only a new starting point to guide experimental studies and design new experiments. And they can help you understand the effect of disease-related mutations, for example, and potentially starting point for drug design. So you can do docking purely ab initio. So without any information, try to predict how two molecules bind to each other. But in real life cases, very often you know a lot about your system. So there might be experimental data, not per se structural data, but it can be just mutagenesis, deletion of domains of your proteins that are telling you which domain is important for binding. So this is interesting information. And this information very often is not used. So if you can incorporate it in some way in your search for the solution, it allows you to drive the docking process. Or if you want to use still more ab initio type of methods, you can use the experimental information as a filter a posteriori to select models that are fitting your data. So over the last 10 years, a bit more than 10 years now, we have been developing HADDOCK, which stands for high ambiguity-driven docking. We can deal with different types of molecules, protein, peptides, nucleic acids, small molecules, and HADDOCK is one of the few methods which can deal simultaneously with up to 6 molecules. In principle, we could push that limit to higher level. It's not really an implementation limit, but you have to realize then when you start dealing with more molecules at the same time, that the complexity is increasing exponentially. And we can also use experimental data. I'm going to explain a little bit more in the next slide to really guide the process. So you're going to use the web server today in the tutorial. The code is available if you want to run things locally, and there is a web server where you can run the docking. So today we're going to make use of one of our servers for that purpose. And one of these servers is also running on a grid, as I explained you in the first day. So what kind of information is typically useful or typically used to look at interactions? We started all the development of HADDOCK initially from NMR data. I'm coming from an NMR lab in Utrecht. And NMR is very good at looking at weak interactions. So even when you cannot solve the structure of complexes in a classical way, in NMR the classical way will be to measure intermolecular NOE distances. So NMR is good at measuring distances. But for weak complexes, for example, this is difficult. But you can still do experiments where you titrate one molecule into the solution of a labeled protein, and you detect changes in the position of the signal in NMR spectra. And this kind of information allows you to map the binding site of a molecule onto another molecule. So this is used a lot in pharmaceutical settings, for example, to screen also for molecule. Because you know where it's binding, you know that it's binding, and you know where it's binding. So this is giving you interface information, basically. NMR gives you much more than that. There are other experiments that are cleaner, but more complex in terms of sample requirement to map interfaces. What I should say here also is that these kind of experiments where you follow signal as function of the concentration of the reagent that you add to the solution, this is not only measuring binding, this is also measuring conformational changes. So if you have allosteric process where something binds on one side of the protein, and on the back side of the protein something happens, it's going to be seen also in the NMR spectra. So not all the data that you measure, or not all the information is really truly interface. So you are measuring more. And this might cause problem also if you put the data in a modeling process, because you are not putting only the true interface, but you are putting more information in there. So you have to deal with this false positive, basically. NMR is also, if you look down there, it's also very good at measuring actually orientations of molecules. So there are experiments that you can do, and Antonio has been telling you also a bit about that, where you can get anisotropy information, which allows you to align molecules in solution. And this alignment, if you know how two molecules are aligned with respect to each other, it's very useful to build a complex. But we are not limited to NMR in terms of what we can use. You can go simply to the wet lab. Mutagenesis information is also useful in combination with some binding assay, of course. So you do mutations, and then you measure binding. And if it's not binding, you can assume that you have mutated a residue on the surface which is important for the interaction. Of course, it can be in these kind of experiments that you have destroyed the structure and you don't measure binding, but it has nothing to do with really the binding surface on the protein. These days, there's a lot of work going on in mass spectrometry techniques to detect interactions as well. You can do basically chemical experiments where you cross-link molecule, so you add very reactive molecules to your complexes, and this is going to cross-links, for example, expose lysines on the surface. And then you lyse those systems, and you detect the fragments, and if you detect fragments that are connected by this chemical entity that you added, you know that those must be at a given distance in space. And the distance depends on the chemical that you're using for the cross-linking. And these days they are even doing reactions where they don't use really small chemicals anymore, but they are going to what they call, I think, zero-length cross-linkers, where you have chemical groups like aspartate on the surface that might be reacting directly with lysine on the other side. So then you have a very tight distance restraint. But the cross-linking, it's not always straightforward to use this information because the distance might be quite long. The error bars are also quite large, so there is not always a unique solution to the problem. But yeah, the big advantage here with MS techniques, so you're speaking of proteomics approach, basically to look at interaction, is that you require very little sample. So you can do these experiments really on proteins that have been isolated from a gel, and that's very powerful to go to large-scale screening of interactions. I should mention also bioinformatic predictions. So in sequence information, actually, there is also hidden information about interactions. And the simplest information will be to think of conservation. So if you analyze multiple sequence alignment and look for conserved residues on the surface of a protein, so conservation on the surface must be functional, otherwise it will not be very likely to be conserved. And this functional conservation is very often associated with binding sites, or it can be the catalytic site of an enzyme. So there's a lot of developments here also to extract information from sequence, combine that potentially with structural information. So if you know the structure of the constituent, you can also analyze properties of their surface, which can be curvature, charge distribution. So there have been a lot of algorithms that have been designed to really predict those interactions. And this is, again, useful information. We make a lot of use of this kind of bioinformatic predictions in our work when you don't have data at all. You can also use, of course, you know everything about SACs by now. So SACs is giving us shape of complexes, radius of gyration, so this is also useful information. I'm going to talk tomorrow about using SACs for scoring, and cryo-electron microscopy is also giving us access to shape of complexes, and then the game will be to dock molecule into a shape. So you see the experimental data are very diverse in their nature, and then we want to use this information in some kind of energy function that we're going to minimize to try to find a solution to the docking problem. So how do we encode this fuzzy information into some kind of energy function? And that's where the high ambiguity part of HADDOCK is coming in. So we define, so what we have are interfaces that are defined between the molecule. And you see here two colors, red and green. So what we call red residues will be we call them active. These are the residues that you have been identifying experimentally or predicting from bioinformatics as being part of the interface. Since we never detect fully the interface experimentally, we are missing regions, maybe we detect too much also. So we usually select also the surface neighbors of those residues to increase the fuzziness of our definition. And that's important for us. And then we're going to define the distance restraint. For the NMR people, it's probably very familiar. It's the same as an ambiguous NOE distance restraint that we use in NMR. But the distance restraint is between any atom of one of these identified residue on one side and any atom of all identified residue on the other side. So you have, and this, so we're calculating all individual distance between all atoms of these residues and all atoms of all those residues, and we sum them as 1 over the distance to the 6th power. And then you take the inverse 6 root of this distance, and this is giving you 1 distance. And we're going to compare this effective distance to a distance that we define, and the distance we say it should be 2 angstrom. What you have to realize in this summation here is that the distance which is coming out of this summation here is always shorter than the shortest distance that enter the sum. It's just a property of this summation. We don't average, but you take a sum, so this sum is going to grow. The more distances you add, the larger the sums becomes. And when you take the inverse of this growing number, you're getting shorter and shorter and shorter distances out of it. Which is why we can use very short distances like 2 angstrom. 2 angstrom is shorter than the van der Waals, the minimum distance between 2 carbon atoms, for example, which will be in the order of 3 angstrom, the minimum approach distance based on the van der Waals radius. And we define 2 angstrom as upper limit, because this distance is always shorter. If you take, for example, 1,000 distances of 5 angstrom, and you do the mathematics, the effective distance which is coming out is in the order of 3 angstrom. Okay, so the distances that you measure are all 5 angstrom, but the effective distance is 3 angstrom. So it's always shorter. And what this distance do is that if one pair of atoms here are getting close, the restraints that we define will be satisfied. So it does not matter which pair, as long as one is getting in proximity. And we define these restraints for all of those residues that have been identified. So we have a network of ambiguous distance restraints that are basically bringing the interface together without defining which contact should be made. So the contact can be ambiguous, the orientations are ambiguous, but we bring the interface together. And by minimizing an energy function, like a quadratic function, which is this distance minus the reference distance that we define, you can basically try to find a minimum to your search problem. Since the information is usually not perfect, we typically randomly delete a fraction of the data. So randomly delete for each docking trial that we're going to do, 50% of the data are deleted. And if we use bioinformatic predictions, this is we go up to almost 90% of the data randomly deleted. For us, it's important because we are driving the search. It's important that the interface be covered. So when you do bioinformatic predictions, it's important to over-predict the interface. So rather, you can make a very accurate prediction, but if you predict only a small fraction of the interface and you're going to force the system to go there, you might force the system to go in a wrong location. You will satisfy this one restraint that you define, but you might get a different interface. So for us, the interface should be part of the definition that we have so that we are able to sample it. And then we delete a large fraction. So the method is energy based, and it's force based also. So we use forces to calculate the gradients, and this allows us to minimize the system, so we know in which direction we should search to find the minimum of the energy function. And the energy function is a classical mechanical functions where we describe bond angles, torsions, and non-bonded interactions, van der Waals and electrostatics. Electrostatics is important to include if you're going to look at interactions. And you add to that the experimental information. So these ambiguous distance restraints, possibly residual dipolar couplings if you have them from NMR or other source of information. And we look for the minimum of this function, assuming that this minimum will give us the right solution. And the search is a combination of energy minimization and molecular dynamic simulations. So we're basically using Newton's second law of motions to let the system evolve through the energy landscape and locate the low energy minima. I will assume that you know that. One of the features of HADDOCK is that we use in part of the protocol torsion angle dynamics. So the degrees of freedom are not the individual atoms anymore, but there are the torsions around bonds. And this is so this is implemented among other in CNS, Explore NIH also supports that. And the advantage of that is that it's very easy for us to freeze degrees of freedom and release them. So we are doing some kind of simulated annealing. So we go to high temperature and cool the system. If you were to let everything flexible, there's a high chance that the protein will unfold, because we don't have restraints to keep the structure together in principle, like if you were to do NMR structure calculations. So we only allow flexibility in the region of the structure that are at the interface. And this is something that you can define automatically on the fly. So for each model that you generate from the docking, you analyze where is the interface, and then you release first the side chain at the interface and then the backbone, and refine the system in that way to allow for small conformational changes. And this is basically the protocol. So we start from randomized orientations, and then we perform first a rigid body minimization, which is driven by the experimental information. And this is basically targeting, as you see here, a specific interface, because in this case, based on NMR data, we had defined the interface as being on this side, but you see that the orientation of the second partner here is not well defined. We bind all here, but here we have different orientations. And this is the characteristic of the ambiguous restraints. They bring the interface together, they don't define the orientation. The orientation has to come from the energetic, the shape complementarity, and things like that. This is very fast, so here we sample between 10,000 or 100,000 structures typically, and then we select a small fraction of those, and then we're going to enter the flexible refinement stage, where we optimize basically we optimize the interface to allow for small conformational changes. And you see here it's a simulated annealing process. So first the molecule moves as rigid bodies first, and then the side chains are allowed to be optimized at the interface. And then in the second phase, the backbone becomes flexible as well. And you will see that you have small conformational changes. If you look at this loop here, this one here, it's going to flip over. And this is driven basically by the data that you put in also. And in that way, you optimize the interface, and the energetics will be better as well. There are a lot of docking programs around that you can use to model complexes. One of the characteristics of HADDOCK is that by doing this refinement stage, and at the end we even refine the structure in explicit solvent in water. So Antonio yesterday showed you the refinement with amber for NMR structure, and this is giving you structures of good quality. So when you deposit, or if you're going to go to validation servers, you will not have clashes at the interface of the structure. They are well optimized. They satisfy typical validation criteria for experimental structures. If you use some of the rigid body docking program based on FFT techniques, for example, fast Fourier transformations, you always end up with a lot of clashes at the interface, and you have to heavily refine those models to get rid of those clashes. So if you were to run validation program on those rigid body or this rigid body FFT docking based structure, you will get a very long list of problems at your interface. And this is taken care of here. So at the end, we cluster the solutions based on typically RMSD clustering or fraction of contact. And then we measure the energetics of the different clusters. So we measure the intermolecular energies of the clusters. And this is going to define our scoring for the different clusters. So we have different levels of flexibility, implicit flexibility. We can start the docking not from a single structure, but from an ensemble structure. So if you have an NMR ensemble structure, or if you have samples from molecular dynamics conformations, you can give those. And the explicit treatment, I explained you already, semi-flexible refinement with side chain and backbone flexibility. So this is the energetics. So this is how we are scoring the solution at different stages. And the main terms are van der Waals. This is a Lennard-Jones potential, electrostatic, a dissolvation term. So this is basically an empirical term which depends on the solvent accessible surface of atom. And whatever if you bury, so if you bury hydrophobic groups, usually it's good because you are releasing water into the bulk and you are winning entropy. So this dissolvation term is a bit of an entropy term. If you bury charges, usually it's not good because you have to dissolvate those charges to bury them, and you are going to pay a penalty in doing that. So this is an empirical term that was developed by Fernandez-Recchio. And in initial stage, we also use the buried surface area. So how large is the interface that you create as a scoring scheme? And the scores have been optimized on a benchmark of system that we are using. And they are not heavily optimized. You see these are round, simple numbers. You could probably optimize further, and instead of having 1, you would have 0.987. But I don't like 0.987. I like simple numbers. And we use the same scoring for nucleic acids, protein, peptides. We have been re-optimizing for nucleic acids, for example. At the end, we are converging to the same scoring. And this is one advantage, because if you have mixed system, you are still using a single scoring function, which is simple to score your system. So you don't need to switch from one to the other, depending on what you are doing. Good. This is the initial complex we were working on at the time, where we basically developed HADDOCK. So this was the complex that was at the basis of the ID. And you see typical NMR data with sequence versus displacement in the spectrum, chemical shift difference, perturbation. And you see that NMR can measure in specific region of the structure perturbation based on due to binding. And then we are translating this information into ambiguous restraints to drive the docking. And at the time, we were ending up with 2 sets of solutions. If you look at those solutions, they are 180-degree rotation of the second molecule, the small molecule. This is a ring finger domain. And if you look at the contact, they both make sense. You find nice electrostatic complementarity in both cases. The scores were similar, so we could not really make a choice between those. But this was instrumental because we had identified important amino acid, potentially important amino acid based on the docking models. So then we went to do the mutagenesis and binding assays, and we could create a double mutant. And you see it here, this guy here, where we have swapped a glutamate for a lysine on one side and a lysine for a glutamate on the other side. And if you have the single mutant, it's not binding anymore, but if you have this double swap mutant, you restore binding. And this is telling you that this combination of amino acid must be interacting, otherwise the double mutant will not be binding. And this corresponds to this model. So again, here an illustration that the model is not the end, but the model is a starting point to create new hypotheses and test them using experimental information. So where does the model stop and when does the structure start? It's not well defined. If you go to PDB, they will not tell you how much experimental data do you need for a structure to classify as structure. So this is just to give you an impression. What we have here on the x-axis is the RMSD from the reference structure. So we know the reference structure. In this case, it's an NMR structure. On the y-axis, it's the score of HADDOCK, arbitrary units. The color coding is black, rigid body docking, red, semi-flexible refinement, first stage of the refinement, and green, refinement in explicit solvent with water. So the 3 stages of HADDOCK. So we can already see is that by introducing more flexibility, by introducing solvents, your energies are going down or your scores are going down. So this is better. So the flexibility does help in refining your structure. if you look at your rigid body models, those guys here, it's not, there is a little bit of correlation with RMSD, but very little. So it's difficult based on rigid body scores only to really select the best structure. But once you add flexibility into the system and you refine system, you see that the correct solution starts to drop and you see those energy funnels appearing. In this plot here, we have only used interface information from NMR data to drive the docking. So we don't have orientational information, but just interface. And you see that we end up with basically two sets of solutions, one funnel here and one funnel here. And these are, again, 180-degree rotated solutions. So we often end up with symmetrical solutions when we do this kind of modeling. So nature is fooling us with symmetry, basically. This one is slightly better. So, and this is so it scores slightly better than this guy, and this is the correct solution. And you see this is the top 10 models of this cluster here. So you can get an idea of the precision of the structure that you get. If you can add a bit of anisotropy information that will define the relative orientation of your molecule. So these are RDCs from NMR. This is also orientational information, but coming from relaxation rates from NMR. You see now that with RDCs, and it's a single set, only the native-like solution really give rise to a strong energy funnel, and it's much more populated than in this case. We still have a few false positive here, and these are still, again, these 180-degree rotated solutions. One set of RDCs does allow for symmetrical solution. But now the energy gap difference is it's much larger, so it's easier to identify those solutions. And you see here the kind of precision that you are getting. The same with relaxation rates, but relaxation rates in NMR are a bit trickier to handle because they are also sensitive. If you have a little bit of aggregation in your system, it's going to affect your rates. And it's going to affect the precision and accuracy of the information that you can extract from NMR. And if you can measure a few intermolecular distances, NOEs, then you are down to doing a classical structure determination by NMR, basically. But you can still do this kind of docking if you know the individual component, and then you see you get only one set of solution, and this is a typical NMR structure. So you can run HADDOCK locally. You can also use the web server. So if you see red colors, it means that it's running locally on a cluster in Utrecht. If you see a blue color of the server, it means that you're going to use the grid version of the server. And for that one, you need to register separately. So you can use the single sign-on mechanism in the WeNMR VO. For the time being, you need a certificate to use the grid version, and we might relax that in the future. And you will see this afternoon that there are different levels of access to the server, from very simple, we just give a list of amino acids and the docking is going to proceed, and more guru interface, basically, where you can get access to more than 500 different parameters that you can fine-tune if you know what you are doing. But here in this one, for example, you can use RDC, residual dipolar coupling for NMR. And if you need access to the more advanced interfaces, you simply have to request it by email. So by default, you get the basic interface. So if you look into the PDB, and this was January, you find about 100 entries which mention HADDOCK as software for the structure determination. So if you look at what kind of information has been used, we have not systematically looked at the data that are there, I must say. Some of them are classical NMR structures where people have been collecting intermolecular NOEs and doing the calculation using HADDOCK, because they are not always easy protocols available out of the box to calculate complexes by NMR, so you can use HADDOCK for this purpose. And you can do protein DNA, protein interfaces you will find here, and protein small ligands, so you find a lot of different things. Some of these probably are also based only on chemical shift perturbation data. They are still made it in the PDB, and you can ask yourself, is this a model, is this a structure? Again, there are no clear distinctions. I think there is a PDB task force, validation task force, who is starting to realize that we are getting structures now that are different kind of information that went into them, so what should we do with them? So they are starting to think about it. So let's move to the modeling of larger assemblies where we have more than two constituents. So I told you already that we can handle the simultaneous docking of up to 6 molecules. There are other softwares that can deal with assemblies and symmetrical assemblies. Most of them will be building the complexes by doing pairwise docking, and then you build from, or you do incremental docking. You dock the first 2 molecules, and then you add molecule number 3, and you do a docking, and then you add number 4, and so on. When we are dealing with multiple components, we do docking of all molecules at the same time. So we don't do incremental docking. You can deal with hetero or homo-oligomers, and important also, you can impose symmetry operations between the molecules and within the molecule as well. So just to show you some results, we tested the method on a small benchmark at the time, and with different type of symmetries. C3, D2 is a combination of C2 symmetry, C5, C2. These days we also support C4 symmetry in the latest version of HADDOCK. It's not yet in the web server. We are updating the web server and hope that in a number of weeks from now we will have a new version of the server that encodes all of these together with some other new stuff. So how do you deal with symmetry? The thing is, you want to have a symmetrical solutions, but you don't know where the symmetry operation is when you do docking, because you're going to get different solutions. The problem has been solved a long time ago by Michael Nilges in the 90s already, who has developed ways of calculating symmetrical homodimeric structure from NMR data. And we use for that the concept of symmetrical distances. So if you have your 2 chains, you can define pairs of distances. So this will be B to X prime and B prime to X. So these are symmetrical distances. And if you define an energy function that's going to tell that those distances should be equal, or the difference between the distances should be 0, and you define an entire set of distances by walking along the sequence here. So you define B to X prime, and then you take the next one. So you define a network of distances of symmetrical distances. And all the symmetrical distances should be equal. This is going to enforce C2 symmetry. It does not matter what the distance is. The distance can be 100 angstrom, the distance can be 5 angstrom. As long as the pairs of distances are equal, this is going to enforce symmetry. And it's not depending on a specific symmetry axis in a system. And that's the way that you can basically build symmetry restraints. There's another type of symmetry which is also used in crystallography, and which is also implemented in CNS, is what is called a non-crystallographic symmetry. And think of that as an RMSD restraint. You say I have 2 molecules that should be identical. The RMSD between them should be 0. So the C2 symmetry restraint is typically imposed, in our case, on the C alpha atoms of the backbone. So this makes that you have symmetrical solution. And we use NCS symmetry to make sure that, for example, side chain arrangement at the interface will be symmetrical as well. So the conformation of the side chains should be symmetrical as well. And using this concept, you can extend basically to different symmetries. For example, if you want to impose C3 symmetry, you're going to define 3 pairs of distances. So for C3 symmetry, AB should be equal to BC, BC should be equal to CA, and CA should be equal to AB. And if you again define this network of distances along your backbone, in that way, this is going to impose a C3 symmetry. And you can do the same for C5. Now you need to define 5 pairs of symmetrical distances. D2, 6 pairs of symmetrical distances. C4 is also implemented now. So a bit of terminology. So how do we measure how good our predictions are? And you can think of all kinds of metrics for that. What we are using is the metrics that the Capri experiment is using. So Capri is this critical assessment of predicted interaction, this kind of international competition for docking. And we have 2 metrics, actually 3 of them, but there is an or in these last 2. So the first one is the fraction of native contacts. So if you have your reference structure, typically a crystal structure, and you calculate all the atomic contact at the interface, and then you measure in your docking model what is the fraction of the contact that are correctly reproduced in the model. So to get a high accuracy prediction, you need to reproduce at least 50% of the atomic contact correctly To get an acceptable solution, you should reproduce at least 10%. Doesn't seem like it's a lot, but the problem is quite challenging. The other metric is a classical RMSD-based metric. And let's look at this one, the interface RMSD. So the interface RMSD is the RMSD that you calculate only on the interfacial region of your complex. So based on a reference structure, you're going to define what is the interface. And the interface is typically all amino acids that are within 10 angstrom from the partner molecule. So this defines the interfacial region, and you calculate the RMSD only on this region. So this is giving you smaller RMSDs, and then you have a cutoff of 4 to get acceptable, 1 to get high quality solutions. The ligand RMSD is a different metric. In the ligand RMSD, you superimpose the structure on the largest of the two components, what we call the receptor, and you calculate the RMSD on the ligand. So you fit on one, you calculate on the other. So this is giving you much larger RMSD, and here the cutoff is 10. So none of those metrics is basically using the complete backbone of the two molecules, because this is not a very good measure. It will change as function of the size of the system. It gives you lower value in general, so people might like it, but it's not a good measure of quality. So we like the interface RMSD in particular, because you are really measuring how good your interface is. If you're docking a cigar, you might get the interface very perfect. But if your cigar is moving like that, the ligand RMSD might be very large, but still the interface is correct. So ligand RMSD is more a global measure. But if you want to do docking because you want to do mutagenesis at your interface to identify important residue, I think getting the native contact is more important than getting the ligand RMSD very accurate. So it depends a bit on what the purpose of the modeling is. Of course, if you want to do docking and then fit into a cryo-electron microscopy map, I think the global RMSD or the ligand RMSD should be right, because if your cigar is moving, then it's going to stick out of your map or not. So these are the metrics. Now, if we did the docking on this benchmark here, and you see the results, so this was actually a Capri target. This was the first Capri target that we predicted. And for that one, since it was a trimer, we had to develop the multi-body components of capability of HADDOCK. And we were one of the few groups that predicted that one correctly, and you see a 2 stars medium quality. The other ones are all high quality predictions, but well, this is also a 2-star protein DNA. These two are docked from free-form structures, so you have the free form of the protein and you don't know the complex. The other ones are docked from there is no free form of the structure. These are obligate complexes. It means they only exist in this oligomeric form, so it's a bit of an easier case for docking. What you see here, C-port is a bioinformatic predictor that we developed. And so we have been using here for driving the docking process bioinformatic predictions. And for the protein DNA one, we were using mutagenesis data, footprinting experiments for the DNA side, so experimental data. So you see this is working. So now what about conformational changes? So we can dock more than 2 molecules. And we can actually use the same concept now to cut molecule in pieces if we expect conformational changes, and then dock the component of the molecule. So we have again a multi-body problems, but just to deal with large conformational changes. So the type of conformational changes that you can expect in when molecules bind to each other are local changes. So they are small loop reorientation, side chain reorientation. You can usually deal with these small ones by docking potentially from an ensemble of structures, ensemble of conformation from NMR, or sampling conformations from molecular dynamics or some other method like elastic network, and using flexible refinement as we are doing. And then you have global changes, and here there are different kind of motions. So you have large-scale domain motions, and this is one illustration here. So you see a protein in its free form and in its bond form, so free in solution or in a complex, and you see that one region of the protein is undergoing a large conformational change, but the domain themselves are not changing that much. So these are global domain motions. So for these kind of conformational changes, you can follow this divide and conquer approach, so cutting the structure in pieces and doing a multi-body docking. And then you have the third class of conformational changes, which will be the binding, induced, folding, or unfolding events. And these are very challenging, because if you don't know a priori that something is changing its conformation, it's basically impossible to predict by classical method, docking methods, to get these kind of things. So a peptide is folding in an alpha helix, or a fragment of protein is folding in an alpha helix when it's binding to its partner, and you have no indication that this is happening. There is basically no way that you're going to model that. For short peptides, you can play trick with that, and I'm going to tell you something about that tomorrow if you're interested. So again, in developing methods, you need benchmarks to validate those. So we put together a benchmark of systems where the receptor was undergoing large conformational changes. So we know the free-form structure of the protein, we know the complex. The ligand here was not showing large conformational changes to limit the problem. And you see the largest one undergoes almost 20 angstrom conformational changes upon binding, and the smaller one on the order of 1 or 2 angstrom. In the docking community in Capri, everything about 2.5 angstrom is considered challenging. So you see these are all challenging systems. So how are we going to do the molecule? So we're going to treat the molecule as a collection of subdomains, and we're going to put restraints between the domain to connect them. So the first thing to do is to define the hinges. So where are the regions where the motion takes place? Okay. Once we identify these, we cut the monomers there. We define interaction restraints between the different domains and add distance restraints for the peptide bonds, and we do a docking. So to predict where the hinges are, we use hinge plot, which is a hinge prediction program. It's a bioinformatic prediction. It's using an elastic network basically to look at the mechanics of your molecule and define hinges. And actually in the tutorial this afternoon you're going to have a look at this. So you cut at the hinge, you have your domains, you have to define your restraints. Of course you don't know which interface interacts with which domain, so you have to define the interface as ambiguous to both domains. So that's why you get those systems. And we add additional restraints to bring back the protein together. But in the initial phase of the docking, you don't want to have very tight restraints because you want the conformational changes to be possible. If you put the peptide bond distance from the start, you're going to reconstitute basically your initial structure, not allowing conformational changes. So we start with loose distance restraints in the order of 10 angstrom. And then later on in the refinement stage, so when you go into the water refinement, you can reduce this distance to something which is more reasonable, like peptide bond. We want compact solutions, so we impose center of mass restraints so that the solutions are compact and you don't get a chain basically. And we do our simultaneous free body docking using the capabilities of HADDOCK. Of course, there are small tricks that you have to worry about. For example, by default, when you use HADDOCK, the termini of your molecule will be charged. Okay, so you get an NH3 plus at the N terminus and COO minus at the C terminus of your molecule. So if you're going to cut your molecule here, you should not introduce charges where you have cut the molecule. A charge difference of 1 can make a huge difference in the docking results that you're going to get. So these are small tricks that you have to worry about when you do that. So here is one case study, and that's the complex that undergoes these 20 angstrom conformational changes. So you see the free form of the protein, so it's kind of a donut shape, and you need to open the donuts to be able to bind the partner. So if you run hinge plot on this system, usually you don't get a single prediction, but you get multiple predictions for hinges. So you have to make a choice. You don't want to cut your molecule in too many pieces, otherwise it's also problematic. So you should minimize. And I think if you are really working with proteins and you have a system, you can do a good educated guess of where things should be cut. There are also things that are unlikely. So if you look at this system, we have 4 predictions. One of them is very close to the N-terminal region. It's not very likely. What is also not very likely are hinge in the middle of beta sheets. So based on work of other groups, in the US, among other, Mark Gerstein has been looking a lot of conformational changes in complexes. You usually don't find hinges in beta sheet regions. You do find hinges in alpha helical regions. And if you remember the talk of Antonio yesterday, he showed one example, calmodulin, where the hinge region is in the middle of an alpha helix, actually. So if you predict hinges in beta sheets, you can usually kind of remove them. And this 1, 2, or 3 actually is in the middle of a loop. And if you look at the system, you can define that there are 2 domains here. It also has the highest B factor in the crystal structure. So that's the one we selected. So you do your docking, free body docking, and you see here that you get a solution which is acceptable. It's far from perfect. You see the superposition. In gray is the crystal structure, and in colored is the docking model. In driving this docking, we assumed that we knew the interface. So we don't know the contact, but we knew the binding regions. We wanted to concentrate on the ability of modeling conformational changes here. So if you're going to do that on your system, and you have very little information to drive the docking, you don't have an idea where the binding interfaces are, it's usually not a good idea to try. I think it will be very challenging. You can try, but I would not trust the results very much. So the more complex your modeling is, the more complex your system, the more experimental information or good bioinformatic prediction you should try to get reliable results. Now, if you do that, if you do a classical 2-body docking, these are the results that you get in terms of interface RMSD. So only 4 complexes will qualify as acceptable, and all those will be unacceptable. If you do this flexible multi-body docking protocol, you see that in all cases you get acceptable solutions. So really it helps. And you see here the rank in terms of models, not of clusters, of the models. So these are all acceptable. So one example, so I show you this one as use case. So this is this example. So you see I get a superposition of the crystal structure onto the docking prediction. This one as well. So we get here about over 50% of the contacts are correctly reproduced, even if your RMSD is far from perfect. And the last one is here, about 2.3. Here, 63% of the contacts are correctly reproduced. And I think this fraction of native contact is very important to realize, because if you're going to base experiments on your docking model, it's important to see that your contacts are reliable. So, incorporating biophysical data, well, I've been speaking already about all the data that we can use. So you can use data a priori as restraints. So you're going to basically really drive the modeling process, hoping that your data points you to the minimum. So you don't want to sample the entire landscape of your binding or binding energy landscape, but you are really directing the search. Of course, if your data are wrong, you're going to search in the wrong region. Okay, so you want to really target the search here. The other way of using experimental information is as posteriori. So you generate a large number of models, and then you're going to score them and remove all the ones that do not explain your experimental data. So this will be a filter. So that's an a posteriori measurements. For our method, if we want to use data as restraints, we should be able to define an energy function, and we should also be able to define the derivative of this energy function, because we are using force-driven methods for the search. And if you can have data or energy function that you can define for which you cannot calculate the derivative with respect to the position of atoms. So if you have this kind of problems, and you can think of system like that, using a filter is simpler, because you don't need to calculate forces. You just need to calculate the fit, basically. So let's see one example of basically an illustration of using experimental data to drive the docking. And that's an NMR-based example. It's a collaboration with Annalisa Pastor in London, where we're looking actually at polyubiquitin chain. And proteins can be ubiquitinated, polyubiquitinated, and this might direct them for degradation, depending on the linkage, or it's also a signaling pathway. So ubiquitination is also used for signaling. And it depends on how, so ubiquitin has several lysines that are on the surface available for making those polyubiquitin chains. And you can get lysine 48 linkage or lysine 63 linkage. Okay, and the lysine is basically connected to the C-terminal of ubiquitin. So these are two possible linkages. And there is an enzyme which is cleaving ubiquitin from the substrate. So you want to recycle ubiquitin basically. And this enzyme is Josephine. And in this work, in collaboration here again with Pastor's group, we were interested in predicting or understanding what is the specificity of this enzyme for the type of linkage that you have in the polyubiquitin chain. So the information that we had was coming from NMR. So we knew from NMR that there were basically two binding sites on Josephine from NMR titration data. We know also from the system where the cleavage site is. So the catalytic triad, the amino acids that are involved in the catalysis of the cleavage are known. So these are those red ones. So you should be bringing one ubiquitin C-terminal close to this site if you want to be able to cleave it. So we use this information to define, to do a multi-body docking. So we docked two ubiquitin and we define ambiguous restraints between the C-terminal of one ubiquitin and lysine 48 or lysine 67. So we don't define which cleavage. So you have two possible binding sites, and then the idea was to look which cleavage does allow to fit those data. And if you do that, you see here one solution where we have the 48 linkage and the 63 linkage. And the 48 linkage is able to satisfy the NMR data better, and it brings actually the C-terminal in closer proximity to the catalytic triad. We did not impose restraints to bring it there. You could have put restraints there, but we wanted to have that as an independent data to validate the solutions. 63 linkage, it's not fitting as well the NMR data, and the C-terminal is a bit more remote, although if you put flexibility in this, you can probably bring it toward the binding site. Experiments were done to measure the cleavage capability of the enzyme. So this is number of hours, and these are the cleavage, the reaction product of the cleavage. And you see that if you use, we are speaking of di-ubiquitin chains, so two ubiquitin attached to each other. You see that the enzyme is indeed cleaving the lysine 48 linkage much better than the lysine 63. If you have free ubiquitin, of course, the situation is different because you can put one here, number 3 can be up there, and number 1 can be here, number 2 up there, and number 3 here. So if you have free ubiquitin chain, I think the cleavage specificity does not matter anymore because you can arrange the ubiquitin in a way that you can still bring number 1 close to the active site. But if you only have a chain of 2, then these enzymes become specific for lysine 48. So this was the outcome of the modeling process. Pseudocontact shift. Pseudocontact shift is also something that our Florence colleagues are very familiar with, and this is also useful information for NMR because it gives you basically both distance and angular information at the same time. So how do you measure pseudocontact shift in NMR? This is again the typical HSQC spectrum, proton versus nitrogen. So you see one signal per amino acid typically, a few side chain on top of that. If your protein has a binding site for a metal, or if you can engineer a binding site on your protein, you can attach tags, for example, to that. And you can replace the metal by a lanthanide. What you're going to see in your spectra is a shift in the signals. This is what is called this pseudocontact shift due to the presence of this lanthanide here. So in the case of complexes, so if one of your protein in the complex does contain a metal binding site, or if you engineer a metal binding site on the system, you're going to measure pseudocontact shift which are intramolecular for the protein which carries the metal or the lanthanide, and you're going to measure effects that are intermolecular for the protein which is binding to it. And we know how to calculate those effects based on the position of the metal. So you can use the intramolecular effects to basically position the metal in the binding site and orient the tensor. And once you have done that, you can then use the intermolecular one to position your second molecule and define the complex. And you see here there are tensor parameters that you need to define, and then there is a function of distance and angular dependence there. So it's giving you basically in one measurement, one set of experimental data, you get both distance and angular information. And this is in principle all you need to define a complex. So how do we do the docking with this kind of data? So first you need to attach your tensor basically and orient it, attach the tensor to your metal site in a protein. So you basically define distance restraints between the tensor and the metal site to position it at the proper location. Then we use the intramolecular pseudocontact shift to orient the tensor in a molecular frame. And once this is done, then you add the second component and do the docking process. So you use at this stage both the intramolecular and the intermolecular pseudocontact shift to get to your docking model. So we developed and tested the protocol using first simulated data, adding noise to them, and this is the epsilon hot complex. These are realistic synthetic data where the variation in terms of the experimental errors are, the errors that we add to the data are in the order of the errors that you measure experimentally. And we simulated different types of metals here. So we have 3 sets. If you do what we call bond-bond docking, so you just take the complex apart and try to reconstitute it, which is an easier exercise because there are no conformational changes, you see that you are getting nicely one set of solution. Again, blue in this case is water. So we are below 1 angstrom in terms of accuracy here. So this is working very nicely. You can do the same using the freeform structure, and this becomes a bit more challenging because if you look at the freeform structure, there are, on one case, there is a loop which is changing its conformation a lot. So this is a large conformational changes for this kind of modeling methods. And the other side, you see that there is a C-terminal tail, or it could be the N-terminal tail, I don't know exactly which one it is here, which is also changing greatly its conformation. If you look at the NMR ensemble, this is an NMR structure, you will realize that this tail here is not structure. So there are X-ray people here. In X-ray, if something is unstructured, you don't see it in the density, and it's not in the model, it's not in the structure that you deposit in a PDB. In NMR, people usually calculate structure using the entire sequence that they have in a sample. So even if they don't have any experimental information to define a conformation, you will find in the structure the chain. So if you're looking at an NMR model, you should look at the ensemble of structure, so visualize all the structure in the ensemble when you download the PDB file, and look at what those tails are doing. If you realize that the tail is not structured and it's sampling a large conformational space, it's better to remove it for the docking process than leave it there, because it might just prevent a good solution. If you look at the yellow one here, this conformation here, if you leave it in your structure when you do the docking, you're never going to generate a solution that fits your real complex, because it will be clashing with the other molecule. So that's very important. So remove the parts that are flexible and unstructured in your structures before doing any docking, because this is going to hurt you. If you do that, you should also not have a charge termini, of course. So don't put charges where they are not present. So again, you do the same exercise, but now from the freeform structure where you have these conformational changes that are quite significant, and you get again solutions that are of acceptable quality, so they are not far from perfect, but still it's pointing to the right interface, and they qualify as acceptable. So this is working. So then we use experimental data for an homologous complex for which there was no structure yet, but there were pseudocontact shifts that had been measured in Oetting's group in Australia. And if you use those data, you can see that you can generate a solution that looks really like a classical NMR structure. And these are now the experimental data, experimental pseudocontact shift versus calculated pseudocontact shift for 3 different metals. And you see the fit for epsilon on this side, and this is the fit of the data for the theta structure, and they fit perfectly. And this is again an unbound docking. You start from the freeform component or from the crystal structure of the freeform, and you generate the complex using the pseudocontact shift only. And this is giving you basically high accuracy structure. So this was described, published already. So, we have about 25 minutes to go before lunch. 3 topics. Think about it. I'm going to drink something, give you some time. So I have protein DNA modeling. I think last year we were here also at ISGC and we gave a workshop about protein DNA modeling, so some of you might have been hearing that. I can talk about small molecule docking with HADDOCK. We're not going to do 10,000 million compound screening because we are not as fast as AutoDock. Or I can tell you something about our adventures in Capri, which is this critical assessment of predicted interactions. And if you choose quickly, we might be able to do maybe 2 topics before lunch. We're going to vote. So who is for? Protein DNA was done yesterday, so let's start with the middle one and then we see where we stand. Small molecule docking. And 1, 2, 3, 4, 6, so that's not so much 7, okay. HADDOCK's adventure in Capri. That's okay, that's more. Who wants DNA? DNA is losing, okay. So we start with Capri and then we see where we stand. So we still have a chance to deal with some of the other stuff. So HADDOCK's adventure in Capri. So Capri is this critical assessment of predicted interaction, and the website is here. So if you want to start participating, actually it's open to all groups. There are actually 2 categories in Capri these days. One is the prediction category, where you have to submit 10 predictions for a complex, and you typically have 3 weeks to do the work. You submit 10 models, and your number 1 model will be the one that you trust the most, typically. And in Capri, there is also a category these days which is about scoring. So all the predictors are asked to contribute models. So they ask to deposit 100 models. Those models are then anonymized, all put together, and basically you are getting a few thousand solutions, and then you have to do the scoring. So based on the generated models by different methods, different groups, try to predict which one is a native-like solution. So if you're interested only in scoring and energetics, Capri is also interesting for you. If you are interested in development of docking methods, you can also participate, and you of course can do both. So we've been participated, well, these are the results for round 4 to 19, 27 targets. So round 4 was the one where we started with this trimer I've been showing you. And since we are information driven, we need to derive information to do the prediction. So we have been relying on both bioinformatics predictions, but also literature. So we always screen, yep.
Do you have time to describe a bit more about the one, how you get the information from literature?
This?
Yes.
Google. Now you need, so here we have 3 weeks. So basically, if you know the complex, so you know the protein. The Capri targets, there is no information available. The structure are usually not published, of course, otherwise it's not a Capri target. And even if a PhD student has put a nice picture of the complex on his website, and the target will be cancelled. So we don't even want to have information about that. But you know the protein, you might know the function. So if you search in literature about that, and you find papers, so it means that it's not automated. So the literature part, you cannot really, although there are websites that are data mining literature, so you can give a word and another word, and if you find the same words in the same sentence, maybe they are related. But what you will find in literature, for example, is, if this residue is mutated, it's not binding anymore. For us, it's very useful information. What you also find in literature are deletion information. People are doing domain deletions, so they are starting cutting parts of their protein until it's not binding anymore. So if you know that the first 60 amino acids are important for binding, and if you delete them, it's not binding anymore, for us, it's useful information because you know that you can target your search to that domain and neglect the remaining. So this is the kind of information that you will typically find.
So how many papers you have to read and I mean, check how long to get enough information for you to publish
So it's really a question of searching. So we have people in group that are very skilled at finding information, I think. So it's not like you're not reading 100 papers. So typically, I think any complex, there's usually not so many information that you find. So, but you might find 10 papers for one target where there is something interesting, and you have to screen quickly for the paper, is there something or not? And then look more into details. So it depends per target. And some targets you don't find anything because it was a target selected from some structural genomic project where they select targets which have no homologies with whatever is known, and then you don't find anything. But it's from target to target, it's different. And then the bioinformatics, again, the same thing. So we use typically our own tools for bioinformatic predictions, but we use also tools from others. And it depends on the amount of sequence information that you can find. So if you cannot generate enough sequence alignments, then most bioinformatic methods will not give you anything. Okay, if you only have a sequence for human and mouse, you cannot measure conservation. So you need enough evolution in the system to be able to start measuring conservation. Okay, so if everything is conserved, then you cannot detect what is significant from what is not. So this is a summary of the of our performance in round 13 to 19. So this was evaluated in the last Capri evaluation, and there was a special issue of Proteins published in 2010 about this. So the blue ones are the ones where we were successful. You see here our number or 10 models from 1 to 10. 1 means it's a high accuracy prediction, so below 1 angstrom, more than 50% of the contact, 3 stars basically in Capri. 2 will be medium accuracy, below 2 angstrom RMSD, interface RMSD, and 30% of contact. And 3 will be, this is the number of stars. So 3 will be high accuracy, 2 will be medium, 1 will be acceptable. And you see also the ranking basically. So you see not everywhere we have the best solution on top. In most cases where we were successful, actually our ranking was not bad except here. So here we selected something else on top. You also see generally that if we have acceptable solutions, we have more than one. So we tend actually to, we're not spreading all solutions to submit 10 models that are completely different, but typically our top 4, top 5 models come from the same cluster. So we tend to trust our data or prediction more. So we are not really spreading the eggs in different baskets. And what you see here are the type of targets. So if you see a U, it's an unbound, so the free form was available of the protein. If you see H, it means that you have to generate an homology model. So you are being measured both on your homology modeling capabilities and docking capabilities at the same time. And if you see a B, it means that they gave us the structure of the bond form of the complex, because there was no free form of that protein, or it could not be modeled based on another structure. There are less and less of those targets now in Capri where they give us the bond form, because it's an easier problem. You have no conformational changes there. So you can basically freeze the protein. So we're also participating to Capri with the HADDOCK server. So here the prediction has to be automated. It does not mean that we cannot input information into the server, so we are still doing our bioinformatic predictions, but the server typically only have 1 day to do the prediction. So once you download the targets from Capri, within 24 hours you should submit your prediction for the server. And then the ranking that's coming out of the server is what we submit directly. And here you see that the server was, in 4 cases to which the server participated, it was successful. So now if you look at the statistics, what does it mean? For those rounds of Capri, we got 15 out of 25 targets correctly predicted, which is 60% success rate. And if you look only at the unbound docking, so where there was only all 3 components homology models, it's 62%. And this is as good as it gets. Okay, so we were belonging there in the top groups. What is also interesting, and I'm going to show you more about that, is the wrong solutions in Capri still usually have the right interface. So even if your model is not correct and you were to say, okay, I'm going to do mutagenesis based on this model, there is a high chance that you are going to find a mutation which is relevant for the interaction. So the model can be wrong, you can still probably get something out of it. And that's very useful to direct the experimental work. The most recent round of Capri, and this has not been evaluated yet. There will be the Capri meeting in Utrecht. I'm organizing that one in April, 17th to 19th of April. So these are our predictions in the last round of Capri. And you see here that we have been doing extremely well. And here we are ahead of the competition by 1 or 2 targets, I think. So here we got 90% success rate, and it's quite amazing. The scoring is not always perfect. So you see that there are, for example, those targets, we didn't do that well, but we got solutions that were acceptable, but they are not on top. This one was interesting. So this was a protein binding to a heparin, so an oligosaccharide. So there's also a lot of conformational flexibility in oligosaccharides. So this was the first time. So this is, I think the one Capri target was once a protein RNA complex. This was also tricky. All the others were protein-protein complex, and now for the first time we are kind of a protein, we will not call it small ligands, but protein oligosaccharide targets. So 90% success rate. So going back to the previous evaluation, so we looked at our solutions, and remember we were only 60% successful in that round of Capri. But if you look at what is the fraction of the true interface that you predict, so that's the interface coverage, even in cases where we are wrong, and some of these cases here we did not predict the correct structure, but if you look at for the receptor and ligand on average, we are clearly above 60 or 70% correct prediction. So you can use docking actually to predict interface here. Meaning again that if you're going to base experimental work coming out of the model on these kind of models, there is a high chance that you find something relevant. If you randomly do mutation on the surface of a protein, on average, only 1/10 or 1/9 of the structure is involved in the binding site. That's the statistic based on the non-complexes. So if you randomly select residues on the surface, you have 1 chance out of 10 to finding a correct or an interesting residue. If you do some modeling, some docking, and then you look at the solutions, you increase your hit rate probably to 1 out of 4, 1 out of 3. And this is time, and it is also money in terms of experiments. Yep?
In Cambridge context, were you provided with NMR restraint?
No.
But as I know so far, the HADDOCK needs those kind of information you want, right?
No, we can. So we don't use NMR restraints. We use just predicted interface. Or in some cases, we just use the entire surface, but we don't.
You just blind search all the surface?
In some cases, we do that. So in some cases where we cannot find bioinformatics predictions because there is not enough sequence information, what we typically do is do a first docking where we sample the entire, and then analyze in that way what are the most contacting residues, and then use that as a second round of docking. But in most cases, you can find either some literature data or we do bioinformatics predictions. And then we input those predictions to HADDOCK. So we are by far not limited to NMR data. And I think there are a lot of NMR users of HADDOCK, but there's a lot of non-NMR users of HADDOCK. I think a lot of crystallographers these days are using it as well, and people who are doing EPR, all kind of data can be put in there, as long as you find something. And for Capri, you have no information. You don't have time to do any experiments.
After the docking, usually the components will be rated by the HADDOCK score, right?
Yes.
How do you decide which one is the real, the good ones? I mean, it's just based on the score. Because sometimes the good one is happen to be ranked at 5 or 10, you know.
Yes, so that's, well, you see our ranking, it was quite good in the prediction I showed you. So one of the things is we do clustering and we rank on clusters. So this is already limiting a lot. So if you do some of the software, FFT docking software are going to give you 2,000 solutions. And so you have to look at a large number. When you do clustering, typically in our hands, when we run Capri, even with fuzzy information, we might end up with 10, 20 clusters at the worst case. So we don't have to look at a huge number of solutions. Our scoring function that we use in HADDOCK is quite robust. So we have good experience with it and we trust it well. But you always have to look at those structures, because a number does not tell the entire story. So in the server, we take the scoring as it comes out of the server, but when we do the manual Capri competition, we look at the solutions, and we look at which contacts are made. And the human eye, I think, is still much better than any software to assess if something makes sense or not.
How many will you suggest initially to the magnetic?
Well, I think if you have to look at 10, so you can look at, look at also at the scores. If the scores are all very close, you should look at all your solutions. If your scores are very different, you can probably look at the top 5 or something, but the score is telling you. So you will see if you are doing the tutorial that we have now added to the server a Z-score also of the cluster. So it tells you, is this unique or is this like all others? We tend in Capri to look, maybe we try many things, we cluster everything, and at the end we might be looking at 20 clusters. But we always eliminate, and we always come very close to our scoring based on the HADDOCK score at the end. But I like to look at the models. So I think you cannot do structural biology or modeling without looking at structures. Don't trust numbers. Look at the structures. If I see nice packing of aromatic residues across the interface, it gives me a good feeling. It might not translate always in a nice number on paper because the energetics are limited. But the model should make sense. And one example of that, the danger of not looking, in one run of Capri, when we were playing with flexibility, we played some tricks, and we ended up with a solution which was ranking fantastic, way better than everything else. And if you don't look at it, you will say this must be the solution. And you look at it, what we had made, we had created a knot. Basically, there were 2 loops that were interconnecting, so you could not separate the protein anymore. But because we do flexible refinement, we refine this perfectly, so this generates a huge interface. The van der Waals energies were very large. So this was ranking fantastically, but it was complete crap. And if you don't look at it, you will not see it, because you cannot detect knots very easily. So one of our weaknesses, so when you run Capri, I think what is interesting also is that you learn of your mistakes. So when things go well, then you're happy and you celebrate, and when you fail, then you go back and try to understand what went wrong. So one of our weaknesses is that we are information-driven, meaning that if we put the wrong information into the system, you're going to get the wrong answer. So that's the danger of information-driven docking, because you're never going to sample the right solution. This was an enzyme inhibitor complex. A lot of groups got that one right, and we got it wrong. We were limited by time, I think, at this time, so we didn't have much time to do things. So we did bioinformatic predictions. The red amino acids are the bioinformatic predictions. And this is what we had to predict. Our structure was here in the middle. Once the crystal structure was released, this is what was in the crystal structure. Okay, so you would see that actually there was more than that in the crystal structure because there was another enzyme up there. So this loop is the inhibitory loop for the other enzyme, and this loop is the inhibitory loop for this copy here. So the stoichiometry is also important. Okay, so you have, if you look now at the interface, the bioinformatic predictions kind of cover both interfaces. So if we had known that this was the case, and we do a free body docking now, a free body docking would have generated solutions that would have been acceptable, or actually even medium quality solutions, because then you block part of the interface with another molecule. So this is also, so you have to worry in any modeling also about the stoichiometry of your system. And you see that doing the simultaneous docking is advantageous because you block regions that will not be blocked if you do a two-body docking. So if you do your incremental docking, in our case, we ended up here in the middle, and if you want to try to add another molecule to that, it's never going to come at the right place because part of the interface is already blocked. So the information is important. Of course, if the information is good, then you're going to get very good models. And this is another target where there were actually 2 binding sites, and here we were a bit smarter in the way that we identified the binding sites. We realized that this is a classical enzyme inhibitor complex, and we found an interesting paper actually that was explaining that the inhibitory loop in this kind of complex has a very specific 5 psi angle pattern in the loop. So knowing this, we have been scanning this unknown structure for 5 psi angle patterns and identifying where the binding motifs were. And doing this scanning, we realized that there were 2 loops that qualify as potential inhibitory loop. And we did 2 sets of docking, and these are the top 5 structure of those 2 sets. And these were 10 structures that were of high quality. And this was the first time that this was happening. Another example, more challenging one, this one. We had to build an homology model for this coil system. In a PDB, there is only a short fragment, so first we had to build this long fragment. And the other problem here is that there are many repeats in this sequence. So in terms, you could shift your protein slightly, and you will find almost the same binding site. Still, we were able to identify the correct one, so energetics was good again, but we have been sampling here all possible binding sites and then scoring them. So we submitted medium quality prediction, and actually for the scoring experiment we had uploaded a high quality prediction, but we didn't select it ourselves. Good. I think seeing the time, we will not speak about protein DNA or protein small molecule. You can look at the slides. The slides are in the handout that you got. And I'm going to move to the last part, the challenges, a little bit. So challenges in the fields. So can you think of using docking of this kind of modeling to predict those interactomes? So we started with the experimental interactome, saying we want to add the structural dimension, but can we turn the problem and say, can we use modeling, given the knowledge of the proteome, of the component of a genome, basically, can we use modeling methods to try to predict those interactomes? So what do we need to do? If we want to do that, we will first have to do the cross-docking of all possible components. This is going to be a large number of docking. Okay, if you take yeast, there's about 6,000 proteins, so it's 6,000 times 6,000, well, divided by 2, so it's but still a large number. So you will need very fast techniques here. In our hands, we will need also to have bioinformatic prediction if you want to do that in large scale. So, but once you have done that, then you still have to identify because if you do docking, you're always going to get a solution out. Okay, you put in a software that those 2 proteins should interact, so the software is going to give you a solution. How do you know if in real life this complex is being formed or not? To answer that question, you will have to predict the affinity of the complex. And that's a very difficult problem because this is coming close to the free energy problem. And free energy is the most challenging quantity to understand and to predict. Even for small molecule, if you do small molecule docking, predicting reading binding affinity is very difficult. I think in small molecule docking, you can do reasonably well if you look at a series of compounds that are very similar. So you take cholesterol-like molecules, and you do small modifications by changing 1 or 2 atoms, probably you are fine. You look at 2 completely different drugs, and then predicting the affinity will be challenging again. So, but we can ask a more simple problem instead of going to complete interactome. Given 3 proteins, can we predict which pair does interact? We will think this is a much simpler problem, but this is still a question that we cannot really answer from a computational perspective only. Or origin of specificity. So we asked the question, how good are our scoring functions? And if you remember the different plots that I've been showing you, when I speak of the HADDOCK score, I say arbitrary units. I don't want to put kcal per mole or something on it, because people think of affinity directly. So it's arbitrary unit. So given the knowledge of the crystal structure, can we predict the binding affinity of a complex? So we've been putting together a benchmark where we had the free form of the structure, the bond form of the complex, and binding affinity data. And what we did is to use HADDOCK for those 124 complexes to build the missing side chains, because you have to realize that in a PDB file sometimes you will be missing side chains, and this will be important if you come to binding affinity. and we run the water refinement. We also did some short minimization only, or running the water refinement HADDOCK to HADDOCK to get the score, basically. But we didn't limit ourselves to HADDOCK only. We look at different methods that are out there. If you go to the EBI, you have the PISA server, which is going to predict all kind of thermodynamic quantities, free energy of interactions, but it's also giving you entropy, for example, of dissociation. HADDOCK is used very much also to score complexes, fast contact from Camacho, DFIRE, Z-rank, it's from the ZDock people, Rosetta, so calculating binding energy, and even more that are not in these slides. And basically, the bad news is that if you take all these and you see PyDock, for example, affinities, another method, attract, is there any correlation between the score given by these docking scoring functions? Okay, those functions were not designed to predict binding affinity. These are scoring functions in docking. And binding affinity, you see that R square is about 0. So there is no correlation. Okay, but it's a different problem. Here we are comparing completely different complexes and see if we can predict and get a correlation. When you do docking, it's always the same protein that you look at and you try to distinguish if this solution is correct or this solution is correct. And that's a different problem. You have to realize that. Just to show you some plots for those complexes, binding affinity versus score, it's not very impressive. So HADDOCK is somewhere there. HADDOCK is here. This was the Rosetta score here. So no correlation. You also have to realize that getting reliable experimental data is also tricky. There are many things that can go wrong, and we realize that also. So we've been cleaning the data, and if you take a subset in collaboration with Joel Jana, Paul Bates, and Ziping Wang, of 48 complexes that we trust more, if there is a his tag in your complex when you measure the affinity, but it's not there when you have the crystal structure, the his tag is missing, and it can have a contribution to the affinity that you measure. If you have one mutation in your system because you wanted to make your protein more soluble for crystallization, it might affect your binding affinity again. So you really have to be careful in what you use in terms of experimental binding affinity data, that they should match the structure that you're looking at. And you see, if you use this cleaner set, it looks a little bit better. Rosetta is here. Now it has an R square of 0.29. So that's not, it gives you an R, the correlation R of 0.5. So there is a correlation there, but it has no predictive capability. In the meantime, we have been extending this benchmark, so it was published in 2011. So there are about 144 complexes here of different binding affinities, which we consider reliable, where you have the free form structure and the bond form structure. And this is at the basis of development for predicting binding affinities. So what are the limitations of the current models? I think the quality of the experimental data is one of them that I mentioned already. Ambiguity in crystal coordinates or missing parts in crystal coordinates that might be relevant to predict binding affinity. Conformational changes, very important. What we have been doing and the results I show you were only in this part of the free energy profile. If you look at what is binding affinity is related to free energy. And it's the difference in free energy between the bond state and the free state. So what you see here are two different complexes. You see you have this circle, triangle, and triangle square. They would have the same absolute energy in their bond state. When we do docking and we score docking solutions, we are only looking at that typically. The difference in binding, in free energy of binding of those two complexes is not coming from their bond state. Here it's coming from the free state. Because their free states have different energies, they are going to give different binding affinities. And these are linked also to conformational changes, the difference in entropy in a free state. So I think, and you can also look at the benchmark that we have, because we know which complex are rigid in their binding and which complex undergo conformational changes. If you only look at the complex that are rigid in binding, you see that the correlation that you are getting are much better. So there you can grasp better the free energy of binding by looking at the structure of the complex only. But as soon as the conformational changes are playing a role, you will have to address the problem of the free state as well in your prediction in order to get reliable prediction. Cofactors might be important also if you don't have them there, but they are present in a real system. They are going to affect your binding. Solvent might play a role. And I think this is also very, well, that's what I've been saying. So in conclusions, well, I've shown you different aspects of HADDOCK, which is very versatile. We didn't speak about nucleic acid or small ligands. And I think the main message is when you do this kind of modeling, you have to see the model as a starting point for a new experiment and really not as an end of the process. So it doesn't take that much effort. It's easy to say when you are doing computational structural biology, but it doesn't take that much effort to go back to the lab and really validate your models. And you can get new information that you can put back into your modeling in the next cycle. So in that aspect, this kind of modeling is complementary to classical methods. But there are many challenges. So I mentioned scoring, prediction of binding affinities and conformational changes. It is still challenging. And nature is still fooling us big time. There are many aspects of molecular recognition that we don't understand. And this is the almost last slide. So this is yeast-to-hybrid screening of interactions. These are E2, E3 complexes. So that's ubiquitination again. Two proteins, and you can read which one. If I will give you the reference. So this is yeast-to-hybrid. So when you see a blue dot, it means that something is interacting. These are checks, basically. So these are not really interactions. So this protein has no interactions. So we are screening around 270 different E3 ring domains. This protein has a rich profile of interaction. The only difference between the two is an aspartate to glutamate mutation. And it's not even in the interface. Okay? It's close to the interface, but it's not in the interface. And I think we understand what's going on in the system, but you see that such a small mutation can have such a huge impact. These are not strong binders. We are not speaking of nanomolar binding here. These are weak interactions, typically in the signaling pathways. So we are speaking of micromolar interactions typically. And this is where small differences are going to make have a big impact in binding affinities and in the regulation of things. And nobody is going to try, if I give you a protein, you're never going to try to do an aspartate glutamate mutations if you want to look at interactions. And this actually, this was published last year in PLOS Computational Biology, and what we have been identifying here is that actually dynamic is important. So this particular amino acid is involved in a salt bridge network, which is close to the interface, but by changing the side chain, you are adding a CH2 group to an aspartate. You change the dynamic of the network, of this salt bridge network, and you shift the equilibrium to a more open binding competent state or closed binding incompetent state. So it's not only static structure, but it's dynamic also, which will be important, especially in those weak interactions. Good. There's a long list of people who are not all in Utrecht now, so these are people who have been contributing over the years to the developments of HADDOCK. And this is a picture of the group that was taken last year. So Joao is here. He looks like the boss. And he's going to assist this afternoon in the tutorial. And everyone has been contributing to some of the aspects that I've been telling you. Thank you very much for your attention. This was last Saturday close to where I'm living, so just to see the difference in it's nice and warm here, and it was cold, but beautiful there.